Welcome to the virtual tour of the Molecular Plant Sciences PhD program at Washington State University. The Molecular Plant Sciences program is unique as it is one of 3 interdisciplinary, interdepartmental programs, and that's why it's housed physically in the graduate school here on our Pullman campus. The Molecular Plant Sciences graduate program has over 45 faculty and averages about 40 graduate students per year. I'm really sorry that you can't be here in person for us to show you the amazing work and facilities here at Washington State University. But right now, I'd like to take you on a tour of some of the facilities that are available to the graduate students and faculty that are associated with the Molecular Plant Sciences graduate program at WSU. Here we are in the wheat research greenhouse facilities, and I'm standing in front of a whole bunch of wheat. Wheat is one of the major crops in eastern Washington, and that includes winter wheat, spring wheat, and club wheat. In fact, Whitman County is the largest wheat-producing county in the country. The growth facilities that we are in today are made up of 2 buildings. The most recent one was completed in 2015, and the total investment in these 2 buildings is over $20 million that came from stakeholders such as the Washington Wheat Commission and the Washington Grain Commission, the state of Washington, and the federal government. These facilities allow us to do cutting-edge research on understanding how plants grow and develop, as well as allowing us to develop new varieties that farmers can grow and increase productivity of their farms throughout the Pacific Northwest. Wheat's not the only plant that we grow in these greenhouses. We also grow barley. We do research on chickpeas and lentils and turf grasses, as well as working with soil microbes, nematode research, disease resistance, plant growth regulators, seed increase, and breeding programs. So the breeding programs will often take a paired approach of understanding the fundamentals of how plants grow and develop, and taking that knowledge and incorporating it into developing new varieties. Once we've developed a new variety in these greenhouse facilities, we need to move them out to a farm to do further research. In fact, here at WSU Pullman, we have 5 farms that are within a 5 to 10 minute drive of this greenhouse facility. Those farms range in size from 5 acres to over 300 acres. Some of the tools that are available for graduate students to work with here include double haploid facility, which allows us to speed up our winter wheat breeding by about 50%. We have spray chambers that allow us to mimic the application of plant growth regulators as well as herbicides the same way that a farmer would put them out in the field. We have a Phenomics facility, which allows us to use robots to study how plants grow and develop over time. We have growth chambers that mimic seed germination conditions in the spring or fall. We also have growth chambers that mimic seedlings overwintering out in the field with low light and low temperature. This is actually required in order for winter wheat to flower. It has to be planted in the fall in order for it to flower the following year. Welcome to the Washington State University Compact Plants Phenomics facility. Dominic Schneider is here to show you what this facility can do.
Hi, my name is Dominic Schneider, and I'm the Phenomics Manager for the Compact Plants Phenomics facility. We are one of 4 Phenomics facilities on campus. We are an automated, high-throughput phenotyping platform for compact plants, and it was established with funding from the M.J. Murdoch Charitable Trust Foundation and multiple entities at Washington State University. The Phenomics system includes 4 camera systems and exists within a growth chamber in which we can control light, temperature, humidity, and CO2 for various growth conditions. The platform carries a suite of cameras. 1, we have a pulsed chlorophyll fluorescence imaging system for measuring photosynthetic activity. 2, we have a visible RGB camera for quantifying leaf area and growth rates and the color of the leaves. 3, we have a near-infrared camera that is used to detect change in water content of the leaves. And 4, we have a 3D laser scanner that produces point clouds of plant structure. Using these camera systems, we can uniquely observe both spatial and temporal heterogeneity in plant phenotypes that would otherwise go unnoticed. Our goal is to provide a high-quality phenotyping service for the WSU community and beyond.
Here we are at the Laboratory for Biotechnology and Bioanalysis, affectionately known as the Genomics and Molecular Biology Core. Derek Pouchnick is here today to show you all of the amazing instrumentation and capabilities that can be used by molecular plant sciences graduate students and faculty.
Hi, and welcome to the Laboratory for Biotechnology and Bioanalysis. My name is Derek Pouchnick, and I'm the director of the Genomics and Molecular Biology Core Lab, LBB. The Core Lab, being established in the early 1990s, has evolved through support from the M.J. Murdoch Foundation, the Center for Reproductive Biology, NIH, and WSU as a state-of-the-art facility for services in genomics and molecular biology. Okay, so now I'd like to go and show you some of the actual instruments that we use for our DNA sequencing and that our graduate students will have access to. The first DNA sequencing service or platform that I'd like to highlight is our ABI 3730 DNA sequencer. Our system here is a 48 capillary system. We can run up to 48 samples at a time. These all get placed into a 96-well plate, and being automated, it can continually run and process multiple 96-well plates. This can be used for verifying plasmids or looking at your PCR products to validate your PCR amplicons. So the next DNA sequencing system that I'd like to show you is our Pacific Biosciences Sequel platform. This DNA sequencer is one of the first long-read DNA sequencing platforms that was developed. When I say long reads, this DNA sequencer can generate DNA sequencing reads of up to 100 kb in length. And it's really been great for doing whole genome DNA sequencing. And in order to really put a DNA genome together, you really need those long reads to span the repeated regions inside a genome. The third and final DNA sequencing platform I'd like to introduce you to is the Ion Proton DNA sequencer. This instrument produces short-read DNA sequences, but although the reads are short, it's massively high throughput. We can generate between 50 to 80 million DNA sequences in a short 2.5-hour sequencing run. This instrument's been really good for doing small-scale RNA-seq experiments, as well as doing ChIP-seq.
Here we are at the WSU Pullman IBC Phenomics Center, currently located in one of our many greenhouses. Magnus Wood is here today to explain its capabilities.
Hello, my name is Magnus Wood. I am the Phenomics Manager here at the IBC Phenomics Center. It was one of the first phenotyping facilities established in the United States in 2009. Here in the center, we utilize a system called the FluorCam XYZ. It is a crane-mounted fluorescence and RGB imaging system. It allows a non-invasive optical monitoring of plants in greenhouse conditions. The device is fully programmable. We can repeat measurements throughout the day, multiple times a day. This allows high throughput for getting many repetitions of each plant. For grad students in the department, we can help you design an experiment, perform the experiment, and then interpret the results of the data.
Our next stop is the Vogel Plant Sciences building. Dr. Rick Sharp is going to show you some of the science that happens in these laboratories. I'd like to point out that we have another brand new plant sciences building here at WSU. And that building will house labs with faculty and students that are part of the Molecular Plant Sciences PhD program.
Hi, I'm Rick Sharp. I am a little biased about this program because I'm a graduate. I graduated in 2014 from the MPS graduate program. And the top 3 floors of the Vogel Plant Sciences building houses researchers from the plant pathology department, the horticulture department, the crops and soils department, and houses USDA researchers as well. The research that is carried out here, a lot of it deals with metabolomics, genomics, phenomics, and transcriptomics. And this type of research generates a lot of data. Now, luckily here at WSU, we have a high-performance computing center that's called CAMIAC. Now, the CAMIAC cluster consists of a collection of cutting-edge computers with 132 nodes. CAMIAC provides a uniform user experience to all the computational researchers at WSU. Having a centralized system lowers the barriers to collaboration by providing a space in which researchers across the university can store data, share code, and build communal software.
Here we are at the Franceschi Microscopy and Imaging Center, also known as the FMIC, and home to the very first transmission electron microscope built in North America. This microscope was built here at Washington State College between 1932 and 1937. I'm here today with Dr. Michael Knoblau, the director of this facility and a faculty member in the Molecular Plant Sciences PhD program.
Yeah, welcome to the Franceschi Microscopy and Imaging Center. My name is Michael Knoblau. I'm director of the center, faculty in the School of Biological Sciences, and faculty in the Molecular Plant Science program. So the center harbors several instruments, mostly light microscopy and electron microscopy. And for electron microscopy, you need to prepare your samples because of the interaction of the electrons that you use for imaging and your sample itself. So here we are in the microtome room. So the microtome room is in order to generate thin sections of the samples that you have previously fixed and embedded. So these samples here, these little black pieces, are embedded in plastic, and the plastic is necessary in order to generate thin sections. The sections that you need for electron microscopy or transmission electron microscopy have to be ultra thin. So usually 50 to 100 nanometer, so this is a billionth of a meter. And this is done with an ultra microtome. So you use a knife like this. This is a diamond knife. It has a 4 millimeter sharpened diamond in here. You install it in the machine, and then the machine basically moves your sample up and slices off a thin section of a dimension that you can define. These samples are then caught with a grid, and this grid finally goes into the transmission electron microscope. So here we are finally in the room with an electron microscope. This here is a transmission electron microscope. Pictures that are generated with a transmission electron microscope look roughly like this here. This is, for example, a viral particle. The structure of COVID, for example, of the virus was revealed with transmission electron microscopy. It's the only tool that can do that. And the reason is the resolution. The resolution is about 1,000 times better than in a light microscope. So with these instruments, you can actually see individual atoms. So here we are in the room with one of our scanning electron microscopes. We have 3 of them. Scanning electron microscope, in contrast to the transmission electron microscope, allows you to look at surfaces. In this case, you look more or less, it's not a real reflection, but you look at electrons that are interacting with the surface and more or less get reflected. And so you can see the topography of the surface. You take an embedded sample, the machine takes a high resolution surface image, and then an ultramicrotome in the microscope slices off a section, and then the machine takes the next image. So step by step, it slices through the entire volume, and we can then 3D reconstruct the volume of an entire cell or tissues and so on. So here we are in the confocal laser scanning microscope room. This here is, in contrast to the other machines that you saw before, is a light microscope. The big advantage here is that you can observe the reaction of live samples. So you can take, for example, a living animal or plant, put it under the microscope, and you can observe it while you manipulate it. And this is visualized in this plant here. We can basically measure the speed of information transmission, what intensity do you need in order to trigger a transmission, and so on. This is what we currently, for example, do here with this microscope. For students, we have 2 classes every semester. So the one is a seminar class where I initially give lectures on the background of microscopy, and then students at the end of the semester give a seminar on their project that they did here in the microscopy center. The other class is a lab class. It's a hands-on lab class in the center. And that means you come in with your project from your own lab that you started at WSU. And we discuss with you how you best apply microscopy to it. And then we teach you how to prepare, how to operate the machines. And once you are familiar with the machines, you're welcome to sign up and come in whenever you want. That's not very common, that students get access to million-dollar equipment. But we have a long history of doing that, and we never had any issues with it. Yeah, it is a good education to get early access to these high-end instruments.
That wraps up our virtual tour of WSU campus and all of the amazing facilities that are available to the graduate students and faculty in the Molecular Plant Sciences program. As you can see, it's snowing now. The weather is known to change around here. I'm really sorry that you couldn't be here with us face to face to see all of the amazing things that are available to the program. But I am convinced that when you arrive as a new PhD student, you will be impressed. Go Cougs!